Hello guys, welcome to Molecular Biology series. I'm Vineet, and today I'll be discussing about the bacterial transposons and also their transposition mechanisms. Okay? So if you talk about the transposons, we can also call them as the jumping genes, and they have a tag saying I could turn a developing snail egg into an elephant. The tag which is given to the transposon is due to no, they increase the quantity of the nuclear genome. Okay, if you look at the Drosophila melanogaster, so its genome quantity has increased over 50% during the course of evolution. Okay, so these transposons, what they do is, they move from one location to another location between the chromosomes, and they increase the quantity of the genome, or induce some kind of mutations. Okay, so this is what transposons do in our bodies. Okay, and they are also referred to as jumping genes. Okay, and these transposons were first discovered by the Barbara McClintock. So she was working in his laboratory, and she observed there was some chromosomal breakages in maize nuclear genome, and these breakages, whatever the broken part is there, she observed that that broken part was on the another chromosome. Okay, so that means, whatever the chromosome that is chromosome part is missing, that part is being observed in different location. Okay, so this kind of changing locations, this kind of DNA sequences which are changing in locations, she called them as the controlling elements and jumping genes. Okay, and later uh, her work was recognized, and she was awarded with the Nobel Prize in Physiology and Medicine because of her elaborate work in the discovery of the transposons. Okay, and she also explained the different color patterns in the Indian maize, uh, and also gave a clear cut concept about the hybrid diseases in the Drosophila melanogaster. Okay? So this is what about the Barbara McClintock and her contribution towards the molecular biology in finding the transposons and their roles. Okay, if we talk about the transposable elements, uh, they can also cause the mutations, or they can also increase the genome quantity. Okay, first one, 50% of the human genome is mainly made up of the transposable elements. Okay, either it might be the retrotransposons or the DNA transposons, whatever they might be. Okay, and some organisms, which contains the large genomes. Okay, so they contain 100 to 1000 copies of the transposable elements, just like the salamanders and barley. So these barley plant and salamander reptile, they also contain around more than 50% of the transposable elements, and we actually don't know the function of these transposable elements, and still a lot of scientists are trying to figure out what's their major role inside the genome. Okay, so one thing is they cause the insertions, deletions, and inversions in the nuclear genome, that is probably causing the mutations only. Apart from that, molecular biologists or the geneticists, they use these transposons as vehicles to carry their, what you call their DNA of interest, okay, or gene of interest. Okay, what they do is, they take a gene of interest and they add some tags to this transposons, and insert the DNA of interest into these transposons, and make the transposons to transpose into different locations, so that that gene of interest gets into some region where the euchromatin, where the chromatin is exposed, and a lot of RNA polymerases will bind and transcribe the gene. So this is what happens in the molecular biology or recombinant DNA technology. So a lot of people, or a lot of geneticists also use these transposable elements as vehicles. Okay, so the major role is causing the mutations, and if it is a useful way, if it is in a useful way, so we can also use them as the vehicles to carry our gene of interest. Okay, so these are the major important things about the transposons and the things what they do. And if we look about the categories of the transposon, they are categorized into two types. One is the conservative transposons and replicative transposons. Okay? And this categorization is mainly based on the mechanism how they transpose from one location to another location. Okay, so which we call them as which we call that as a transposition. Okay, uh, no, breaking one segment of the DNA and getting inserted into that segment region is called as what transposition. Okay, so based on this mechanism, we have two categories. So first one is the conservative transposition, where a lot of non-replicative transposons are involved. Okay, so they don't get replicated, they actually excise themselves from the actual position and get inserted into different position on the chromosomes. Okay, so this conservative transposition mainly transposed by via cut and paste mechanism. Okay, so that means they cut themselves from the origins and they get inserted, or they paste themselves in different locations. Okay, so this is what the conservative transposition do, and most of the bacterial DNA transposons or eukaryotic DNA transposons are mainly based on this type of mechanisms. Okay, and then we have the replicative transposons where they copy themselves, like they first undergo the mRNA transcript. They are first undergo the transcription and produce the mRNA, and this mRNA will produce some sort of enzymes like transposase, resolvase, and some RNA binding proteins, and they cut themselves and paste themselves on the some other location, and they synthesize the new strands. Okay, so by using some kind of polymerases, they synthesize the new strands. Okay, so this synthesis is actually called as what copy and paste transpositions. Okay, in the copy and paste transposition, the original transposon is conserved, as well as the new transposition will get acquired in different locations. Okay, so this is what the replicative transposons do. Okay, they replicate themselves. Okay, so they copy themselves and paste themselves in different locations. Okay, this is called as the replicative transposition, and they cause a lot of proliferation inside the nuclear genome. Okay, and both involves recombination processes, and this is how they look like. Okay, so what we have here is the so this one is the transposon. Okay, and if it is getting doubled or duplicated and or getting inserted in a different location, and the original is preserved. Okay, original transposon is conserved. Okay, so this is the original one, and this is somewhat copied one. Okay, so this one. Okay, so we have two transposons after the replication transposition process. Okay, but in the case of conservative transposition, we only see the actual transposon getting excised and placing getting inserted into a different locations. Okay, so only one actual transposon will be present in the conservative one because it is a cut and paste mechanism. So this is what about the categorization of the transposon based on the mechanisms. Okay, then we have different transposable elements, whether it might be in the prokaryotes or eukaryotes. Okay, so eukaryotes have very minimal amount of the DNA transposons and have lots and lots of retrotransposons. Okay, which involves an RNA intermediate for their transpositions. Okay, so DNA transposons and also the retrotransposons are the major transposons which are found in the prokaryotes as well as the eukaryotes. Okay, the first one what we have here is the insertion sequences. Okay, these are the simplest and the what you call them as simplest transposons which are found in the bacteria. Okay, bacteria sometimes they are also present in the eukaryotic organisms. Okay, but rarely seen in the we will be rarely seeing these insertion sequences in the eukaryotes, but mostly this will be observed in the bacteria. Okay, mainly the prokaryotes. Okay, so these insertion sequences they transpose via cut and paste transposition. So that means in the conservative way. Okay, and they also have some kind of repeats at the terminals. Okay, so 5' terminal and the 3' terminal. So these are oriented in inverted manner, so which we call them as the inverted repeats or inverted terminal repeats. Okay, and the length of this insertion sequence are about 750 base pairs to 1500 base pairs. Okay, and then we have the simple transposition, simple transposons, which are also similar to these insertion sequences only, and they are also found in the bacteria. Okay, and the length changes, but everything is same when you compare this in simple transposons with the insertion sequences. Okay, so the length of simple transposons is around 1300 base pair to 5000 base pairs, but they also have the terminal repeats as inverted repeats, and they also transpose via cut and paste transposition mechanisms. Okay, then we have the composite transposons. Okay, which are entirely different from the insertion sequences. Okay, because they have insertion sequences flanked between between the not between the they actually have a transposable element flanked by these insertion sequences. Okay, that means so this is one insertion sequence, and this one is the another insertion sequence. Okay, we have 5' over here and 3' over here, and this entire is a this entire thing is a transposable element. Okay, first main component what we have in the middle is what the major transposable element, which gets inserted into different locations of the chromosomes. Okay, so this transposable element is flanked by two insertion elements. Okay, so that is what makes a composite transposons. Okay, this composite transposons has the terminal reports as the insertion sequences, and they also transpose via cut and paste transposition mechanisms. Okay, and the length, if you look at the length, the length is actually much bigger than the insertion sequence of the simple transposon, that is around 2500 base pair to 10,000 base pairs. Okay, and then we have the complex transposon, which we also call them as the non-composite transposons. Okay, they are much more bigger than the composite transposons. Okay, so the difference between the complex and composite transposons is they involve replicative mode of transpositions. Okay, complex transposons involve the replicative transposition. That means they replicate themselves. Okay, they synthesize the DNA strands. Okay, and then get inserted in into the different locus of the chromosomes. Okay, so this is what the difference between the composite and the complex transposons. Okay, they also have the inverted repeats, not the insertion sequences. Okay, and then we have the bacteriophage mu, which is mainly found in the bacteria, which is actually a viral DNA sequence or viral DNA sequences, which is actually found in the bacteria. Okay, so this bacteriophage mu, one of the family member of the bacteriophages. Okay, so this inserts its DNA sequence during the lysogeny process. Okay, so whenever the bacteriophage undergoes lysogeny process, it inserts the genetic material into the bacterial chromosomes, and it perform the transpositions or the transposition mechanisms. Okay, like it will perform robust transpositions during the lytic cycle. Okay, so that means insertion is happening different stage, and the actual transposition mechanism is happening in the different stage. Okay, so in the lysogeny it will insert its DNA sequence, bacteriophage mu inserts its DNA sequence into the bacteria. Okay, in this lysogeny phase, and in the lytic phase, robust transposition mechanism will be seen inside the bacteria. Okay, so this is what the bacteriophage mu does inside the bacteria. Okay, so this is how it acts as a transposon. Okay, then we have the conjugative transposons, which is a bacterial transposon, a prokaryotic one. Okay, so this transposon, if it want to get transposed, or if it want to perform the transposition, it involves two bacteria. Okay, so two bacteria has to get linked by the F pili. They have to get conjugated, and upon conjugation the transposon will transfer from donor bacterium to the recipient bacteria. Okay, so if you talk about the bacteria, generally whenever they get conjugated, they will be present like this. Okay, this is called as what, a recipient bacteria, and this is what called as a donor bacteria. Okay, so the donor bacteria will be having some kind of transposer element here over here, and it will be the recipient bacteria will be having its own chromosome material. This donor also has a chromosome material. Okay, so during the conjugation, what happens, this transposon, okay, so during the conjugation will enter into the recipient bacteria and gets inserted into the bacterial chromosomes. Okay, this is what called as a conjugative transposon and the conjugative transposition. Okay, the length is very short because it is a bacterial transposon. Okay, so and it involves majorly the plasmids. Okay, so this length of this conjugative transposon is around 30 to 150 kilo base pairs. And then what we have is the mechanism, that is transfer plus integration. Okay, so this also uses a special enzyme called as the XIS enzyme, which is also known as the excision, and it also utilizes INT enzyme, which is also known as the integrase enzyme. Okay, so these two are the major enzymes which are involved in the conjugative transposition. And then we have the integrons and cassets and helitrons. Helitrons are one major eukaryotic transposons, okay, which are rolling circle transposable elements, okay, which you call this, what you call these electrons as rolling circle transposable elements, okay. So these electrons are also called as the rolling circle transposable elements, and the mechanism, which involves, for their transposition process is replicase plus the helicase, okay. So they also involve the replication and also has the helicase activity, okay. Length is also quite comparable to the composite transposons, okay, 5,000 to 10,000 base pairs, okay. And then we have the retroviral, which is a normal RNA mediated transposons, okay, which involves an RNA as an intermediate. And then we have the endogenous retrovirus, which is actually same, where this retrovirus and the endogenous RNA endogenous retrovirus transposons are quite same only. They consist, direct long terminal repeats, okay. These terminal repeats are much more longer than the inverted repeats or direct repeats, okay. So these long terminal repeats are majorly present in the RNA mediated transposons, okay, that is retrovirus transposon to the endogenous retrovirus transposons, okay. And retrovirus like elements are also same as this retrovirus and the second category. And the retrotransposons contains all these things, okay. The retrovirus mediated and all these things comes under the retrotransposon category. They are also transposed by an RNA intermediate, okay. So lines and signs are the two major transposons, which we also call them as a retrotransposon, but they don't have any kind of long terminal repeats, okay. If you look at the lines, they don't have any kind of long terminal repeats, but they transpose via an RNA intermediate, okay. So they also code for an mRNA, and this mRNA will again get converted to DNA by the reverse transcriptase enzyme, and this DNA will get inserted into different chromosome locations, okay. And then we have the retron and retrointron, which are basically the retroviral transposons only. And the retrointron is actually an intron sequence which get transcribed into mRNA, then converts into the DNA and inserted into different locations, okay. So these are all the basic transposons of the the related elements which are found in the prokaryotes as well as the eukaryotes, okay. Then we have the first topic, the DNA transposons of prokaryotes, okay. So in the next lecture we will be discussing about the eukaryotic transposons. For today, in this lecture we will be discussing about the transposons of prokaryotes, okay. So prokaryotes majorly has 7 to 8 different types of transposons, but majorly only 2 or 3 different types of transposons are discussed in college level syllabuses or any PG postgraduate level syllabuses, okay. So there are 3 major important transposons for prokaryotes, okay, which are actually needed to discuss, but I've also included extra 2. So one is the simple transposer element, which we also call as insertion sequences, okay. They only contain the information required for the movement and for the transposition activity, okay. And then we have the composite transposons such as T9 and T10 elements, or transposon 9 and transposon 10 elements, okay. Tn represent the transposon. They contain the DNA sequence which doesn't contain any kind of inverted repeats, but they have the insertion sequence as that repeats, okay. And then we have the non-composite transposons, okay. So we have a Tn21 and Tn3 like elements, that is transposon 21 or transposon 3 like elements, okay. So the major difference between the composite and non-composite is the transposition mechanisms, okay. The composite involves cut and paste transposition mechanism, that is the conservative one, while the non-composite they involve the what we call them as replicative transposition mechanisms, okay. And both composite and non-composite transposons, they also contain an antibiotic resistant gene, okay, which will be getting transposed from one location to another locations, okay. They both have the insertion sequences flanked on either sides, okay. Then what we have is the mu phages. What I've discussed just now is the mu phages, which are found in the bacteria, okay. So these viral DNA sequences first gets inserted into the bacterial chromosomes during the lysogeny process, and then perform the robust transpositions during the lytic processes, okay. Then we have the conjugated transposon, which involves the 2 bacteria for a transposon to get transferred into different locations, okay, different chromosomal locations, okay. So these are the major transposons which are found in the prokaryotes. The DNA transposons, okay. Eukaryotes contain minimal amount of the DNA transposon, but mostly eukaryotes will be containing the retroviral transposons, okay, which has a RNA as an intermediate, okay. So first one is the insertion sequences. So these insertion sequences are the simplest and normal constituent of the bacterial chromosomes. I've said you can know these insertion sequences are present will be present in lots of bacterial species, okay. They are present in lots of bacterial species and they are the simplest forms, okay. And they also contains very short transposable elements which is of 800 base pair to 1000 base pair length, okay. So how these insertion sequences were identified? First thing, so these insertion sequences were identified during the Gall-Aperon studies, okay. So some researchers they were studying the Gall-Aperon, the functioning of the Gall-Aperon, and these researchers discovered there were some mutations, okay, so in expression of these genes, okay. So this mutation doesn't have any properties like properties of point mutations or the deletion, but they have some properties related to the insertion type of mutations, okay. That means these mutations are caused due to the some kind of insertions, okay. And these insertions are mainly mainly due to a DNA segment which is having a length of 800 base pairs, and this 800 base pair DNA segment is getting inserted into this Gall-Aperon at the initial sites, okay. If you consider this long thing as the Gall-Aperon, okay, which has some kind of operating unit over here and some kind of promoter over here, and we have different kind of structural genes over here, okay. So there was a 800 base pair insertion sequence getting inserted at these initial sites, okay, here, okay. So this caused a lot of mutations in the Gall-Aperon, okay. So this type of mutation can be excised from the locations, okay. That means this 800 base pair DNA can itself excised from this location and getting transferred to different types of locations, okay. So during these studies these insertion sequences were observed, okay. So and what we call this insertion sequence now as a transposons, okay. And the promoters which are present within these insertion sequences, okay, within this insertion sequence of the DNA sequences, okay, they also have major property to alter the expression of the genes, okay, which are present in the Gall-Aperon, the structural genes, okay. They also involved in the altering the expression of this structural genes of the Gall-Aperon, okay. So that means these insertion sequence can also cause the mutations while doing the transposition activity, okay. So that is the major thing about the insertion sequences. And if you look at the the structure of the insertion sequence, okay, we have direct repeats on either sides of the transposable elements and also the inverted repeats on either side of the transposable element, okay. So which are present inside the transposable elements, okay. The direct repeats are actually read in one direction like this, okay. These repeats are oriented in one direction. Since they are oriented in one direction, these are called as the direct repeats, okay, or flanking direct repeats, because they are present on either sides of the transposable elements, okay. Then what we have is the terminal inverted repeats which are read in opposite directions, okay. If you are reading this DNA sequence in this direction, okay, the same opposite direction we have the DNA sequence over here, okay. The same DNA sequence which is oriented in opposite direction is present in 3' to 5' direction tail, okay. This strand, we have a 5' to 3' strand over here on the above, and then we have the 3' to 5' strand below it, okay. So these kind of opposite oriented sequences are also called as the terminal inverted repeats, okay. And even if you look at this 3' to 5' strand, we have a insertion sequence over here, we have a repeat over here TAG CTG, and the same sequence is read in opposite direction, or which is oriented in opposite direction, okay, like this TGCGAT, okay. So these kind of repeats which are oriented in opposite direction are also called as the terminal inverted repeats, okay. And transposal element contains the two terminal inverted repeats, and also flanking direct repeats, okay, which are oriented in the same direction, okay, or in the one direction, okay. So this is the basic structure of the transposable element, and we have lot of insertion sequences in bacteria. Majorly IS1 is the major insertion sequence which is present inside the bacteria, and the simplest form of transposon, okay. And these insertion sequences first they encode for an enzyme called as a transposase, okay. So this transposase enzyme is majorly responsible for the transposition activity, okay, and the frequency depends on the type of insertion sequences, okay. And this transposon first mainly cuts the transposer elements, okay, like this. So it makes the blunt ends, okay, and it performs the staggered ends in the recipient DNA molecule, okay. I'll tell you about that, okay, what's the mechanism of the transposase enzyme, okay. See, we have a lot of insertion sequences in the bacteria. The major one is the IS1, okay, which is of 768 base pair length, and it randomly gets into the different locations of the chromosomes, okay. The target selection is quite random, and it has invert repeats which has 23 base pairs in them, and also the target repeats should contain the 9 bases, okay. So if it if this transposon, if it wants to get into some other random locations, okay, so that random location should contain 9 base pair repeats and can able to undergo a staggered cut like this, okay. So this target repeat must contain the 9 base pair repeat to perform the transposition process, okay. Then we have the IS2 which requires a 5 base pair target repeat, and also has the 41 base pair inverted repeats, and the actual length of this IS2 is what 1327 overall length, and majorly the selection is hotspots, okay. Hotspots in the sense the major gene transcribing regions, okay, wherever the gene active regions are there, at those regions this IS2 element insertion sequence 2 element get inserted, okay, on the hotspot in the hotspots regions, okay. And then we have the IS4, IS5, IS10, IS50R, and IS903. All these are insertion sequences, okay, and most of them have the random target selections, and also some has the hotspot as the target selections, okay. That means gene active regions, okay. And the typical insertion sequence, the length of the typical insertion sequence varies from the 700 base pairs or 768 base pairs to around 5000 base pairs, okay. The 5000 insertion sequence is not given here, but the average is what 1531 base pair length, okay. So this is the highest one. We also have the 5000 base pair insertion sequence over here, okay. If you look at this point, this one, among bacteria as a whole, the insertion elements range in size from 768 base pairs to 5000 base pairs, okay. This is what the average length is, okay. And they also encode for a transposase enzyme, okay. And this is the typical insertion sequence structure of a typical insertion sequence, which has two open reading frames. One is the ORF-A and one is the ORF-B, and also has a promoter, and this promoter is responsible for the alteration of the gene expressions whenever it gets transposed into different locations, okay, on the chromosome. So this promoter alters the gene expressions of neighboring genes, okay. Then we have the inverted repeats over here, this one, and we also have the direct repeats. We will be having the direct repeats over here, but this is a typical insertion sequence. We should consider only inverted repeats, okay. So this ORF-A will code for a protein that is also regarded as an RNA binding protein, or a regulatory protein, okay. Will come from the ORF-A, and entire this element, okay, ORF-B and ORF-A together codes for an enzyme called as a transposase, which is responsible for the transposition activity, okay. How does this transposition works? First, it makes the blunt end cuts on the transposable elements like this. These are the blunt ends, okay, where the ends of a DNA sequence are exactly equal, okay. So these we called as the blunt ends, okay. Blunt end cuts are made on the donor molecule, okay, donor transposon element, okay. So this is the donor insertion sequence element, okay, and on the donor insertion sequence element, blunt ends are made by the transposon, okay. So this transposase makes the blunt ends on the donor insertion sequence, okay, and it also makes the staggered cuts in the recipient chromosomal DNA sequence, okay. So this is the recipient ones. Okay, so this is the recipient DNA sequence, and where this transposon makes the staggered cuts. Okay, this is what called as a staggered cuts, where the cuts are of unequal sizes. Okay, a staggered cut is made in the target site, and the insertion element is then inserted. Okay, so then the transposase, after cutting the recipient DNA sequence, it will insert the entire insertion sequence between these staggered cuts. Okay, and then a new DNA is synthesized in these regions. Okay, wherever the staggered cut has occurred, wherever the unequal cuts were performed. Okay, so at that sites, the new DNA gets synthesized. Okay, and the entire insertion sequence get inserted into the new location. Okay, this is what the new location. Okay, so this is what the actual transposition activity of this insertion sequences, where the insertion sequence get excised from the donor molecule, donor DNA sequence excised completely, and get inserted into the different locations. Okay, this is what we call as a cut and paste transposition, or the conservative mode of transpositions. Okay, so this is about the insertion sequences which are found in the bacteria, which are the simplest and the normal transposons. Okay, and then we have the composite transposons. Okay, so these composite transposons, they often carry the genes which encodes for the antibiotic resistance. Okay, that might be the ampicillin, that might be the tetracycline, or streptomycin, or vancomycin, or some kind of antibiotic resistance. Okay, vancomycin. So these composites, unlike these insertion sequences, they have insertion sequences as flanking repeats, which are located on either sides of a transposal elements. Okay, like now if we have a transposable element on here, okay, so the insertion sequence will be present on either sides of this transposable element. Okay, so this is what called as the composite transposons. Okay, composite, or you can also call them as a complex transposons. Okay, so these composite transposons are also found in the bacteria or prokaryotic organisms. Okay, so some composite transposons have identical insertion sequence elements at either sides, and some might have different insertion sequence. Okay, so this might be IS1 and IS1, or this might be the IS1, then you have a transposon over here, and then IS2. Okay, so these are different types of insertion elements based on the these are different type of composite transposons based on the insertion sequence elements. Okay, and the orientation is also quite different. Okay, so if the insertion sequences, if they are oriented in one direction, or if they are present as direct repeats, okay, as in the case of transposon 9, okay, which we call them as the direct composite transposons, or the straight composite transposons. Okay, and sometimes this repeats, okay, insertion sequence elements may also present in the inverted orientation. Okay, so as in the case of this transposon 5 and the transposon 10. Okay, so the transposon 9 contains direct repeats as insertion sequence elements. Okay, and this transposon 5 and transposon 10 contains the inverted insertion sequence elements. Okay, so this is what difference between the two categories of this composite transposons. Okay, and we also have lot of composite transposons, okay, in bacteria. Okay, so mostly 4 of them performs by major transposition activities inside the bacteria. Okay, so first one is what the transposition 9, 10, 5 and 903. Okay, so this transposition 903 will be providing kanamycin resistance, okay, along with the TN5. Okay, so the transposition 5 and transposition 9 will have the kanamycin resistance antibiotic gene, and 10 will be having the tetracycline resistance antibiotic gene, and 9 will be having the chloramphenicol resistant antibiotic gene. Okay, so these are the different types of antibiotic resistant genes which these transposons contains, along with the insertion sequence elements as their repeats. Okay, if you look at the average length, they vary from the 2500 base pairs to 9300 base pairs. Okay, and associated elements will be the IS1 for the TN9, as I said you in this case. Okay, and IS10 for the TN10 transposon 10, and IS50 for the transposon 5. IS represent the insertion sequence, remember. Okay, and IS903 for the transposon 903. Okay, so these are the composite transposons, and their antibiotic resistant genes, and also the length of different composite transposons. Okay, and if you look at the typical structure of this transposon composite transposons, they have the insertion sequences flanked on either sides of a transposon element. Okay, with this transposon element also contains a antibiotic resistant gene, that is, in this case, it is the transposon 10, so that contains the tetracycline resistant gene. Okay, so the entire thing is also called as what, transposon, or the composite transposon. Okay, so this is what basic introduction about the transposon. Okay, and one more thing is, this transposon 9 and this transposon 903 will be having a monomer transposase enzyme. Transposon enzyme will be the monomer. Okay, while this transposon 10 and transposon will be having a dimer as transposon. Okay, this transposon enzyme will be a dimer in the case of transposon 10 and the transposon 5. Okay, for their activities, they require some kind of transposase enzyme. So this transposon will be monomer in the case of transposon 9, and it will be the dimer in the case of transposon 5 and 10. Okay, so how does this composite transposons do their mechanism? Okay? How do they get transposed from one location to another locations? Okay, now the same mechanism which is involved in the insertion sequence, but what happens is, they involve some sort of recombination methods. Okay, so first they make the blunted cuts on the recipient molecule over here. Okay, which is entirely different from the insertion sequences. Okay? And staggered cuts are made on the donor molecule. Okay, so this is the donor. Okay, and then what we have is, this is the recipient. Okay, so in the case of composite transposons, okay, staggered cuts are made on the donor molecule, while the blunt ends are made on the recipient molecule. Okay, and the transposon along with the insertion sequences are nicked, and it is transposed from the donor location to the donor side to the recipient side. Okay, here. So this is what happens in the case of composite transposons, and then the extra sequences will get synthesized over here, and the entire composite transposon is now in the different location. Okay, so this is what the actual mechanism of the a simple composite transposon. But if you look at this transposon 5 and transposon 10, which are also a composite transposon, they will have different kind of mechanisms. Okay, and the transposition of this TN10 is quite rare, which happens in 10 to the power of 7 times per cell generations. Okay, 10 to the power of 7 generations. Okay, that means cell has to divide, continuously it goes on divides. Okay, for like 10 to the power of 7 generations. Okay, after this many generations, we will be seeing only one transposition mechanism for this TN element, TN10 element. Okay, so TN10 normally doesn't undergo transposition. It rarely happens. Okay, its frequency is very, very, very rare. Okay, so when you compare the transposition activity with the insertion sequence elements, these composite transposons, they have quite less transposition activity. Okay, and this activity is much less when in the case of this transposon 10 element. Okay, then what we have is the transposon 5 and transposon 10. Both has transposase as dimers. Okay, these transposanes which are responsible for the transposon 5 and transposon 10 activity. So this transposase will be functioning as a dimers. Okay, and each subunit in the dimer has active site. Okay, so that is involved in the bond breaking and bond cleaving, or ligation properties. Okay, has ligation properties. Okay, so this is how the transposition of the composite transposon happens. Okay, first what happens is, between the insertion sequences, the staggered cut is made. Staggered cut is made on the donor molecule, and here what we have is a target sequences. Okay, so there are two ways where the composite transposons gets inserted into the different locations. Okay, so in one way, what happens is, only some parts, okay, so like this A and B locations, okay, or C and D locations. Okay, so these sequences, okay, which has insertion sequences on either sides, okay, they might get transposed, or only half part of the transposon, actual transposon, get transposed. Okay, from one location to another. If not, complete transposon which has, four sequences, complete transposon get transposed into different locations. Okay, all of A and D moves. Okay, so this is what happens in the case of bacteria. Okay, either half part of the composite transposon moves from donor to recipient, or complete total transposon will move from one location to another location. Okay, so this is what the basic mechanism of this composite transposition activity. Then what we have is the special transposase which is responsible for the TN5 and TN10 elements. Okay, so as I said to you, it functions as the dimers, and both dimers has active sites. Okay, so one more each of the dimer cut the DNA sequences. Okay, so these active sites are majorly involved in the bond cleavage sites. Okay, they cut the DNA sequences, and they then ligate the molecules. Okay, so what happens is, if you consider this TN5 element, okay, first the 3' hydroxyl group, this part is broken down, this part gets cleaved. Okay, so this will be acting as a 3' hydroxyl end. Okay, now, and here we will be having the 5' phosphate molecule. Okay, so this 3' hydroxyl again attacks on the below strand. Okay, so it will attack the below strand's nitrogenous base. Okay, and forms a hairpin loop like this. Okay, and then this hairpin loop is hydrolyzed, okay, into blunt ends. Okay, and these blunt ends will now ready are now ready for the to accept a transposon molecule. Okay, so they will now accept the transposon molecule. Okay, so this is what happens in the case of this transposition mechanisms. Okay, then what we have is the non-composite transposons. Okay, so which are quite different from this composite transposon because they have a replicative transposition mechanism. Okay, they don't undergo cut and paste mechanism. They don't have any kind of cut and paste transposition activity. They only have the replicative transposition activities. Okay, now the major example for this non-composite transposon is what TN3 element called transposon 3, a very well known retrotransposon, or replicative transposon, which is found in the bacteria. Okay, and apart from the transposase enzyme, these non-composite transposons also involves a special enzyme called as a resolvase, which is involved in the recombination process. Okay, to resolve the linked DNA strands. Okay, in the recombination events. Okay, so this transposase catalyzes the insertion of the transposon, while the resolvase, this enzyme, okay, so this is involved in the recombination events which are associated in the transposition mechanisms. Okay, the major one is the TN3, which produces a 5 base pair target site duplications. Okay, so duplication in the sense that must be a replicative process. Okay, so this transposon, non-composite transposons, are completely the replicative transposons. Okay, so that means they get duplicated when they get inserted into the different sequence of different region of the chromosomes. Okay, and we also have a lot of different non-composite transposon, as in the case TN1 and T3. And apart from this transposase enzyme activities and the resolvase enzyme activities, they also code for a antibiotic resistant genes. Okay, so TN1 and TN3 are the complex transposons that carry the resistance to antibiotic of penicillin. Okay, so they carry the penicillin antibiotic resistance activities. Okay, and they are also found in the plasmids and also the chromosomes of many bacteria. Okay, then this TN3 is actually 4957 base pair length. Okay, so its actual length is 497 base pairs. Okay, and we have a very first gene, that is the BLA gene, which encodes for the lactamase. Okay, so which destroys the antibiotics such as penicillin and ampicillin. And then we have the TNPA gene, which code for the transpose enzyme. Okay, so this recognizes the inverted repeats and the target sequences and cuts the sequences. Okay, which is responsible for the cutting the sequences, this transpose enzyme. And the TNPB, which is involved in the resolvase enzyme activity. This recognizes the IRS sequences or the internal resolution sites during the recombination events, and resolve the DNA sequences. Okay, or cut the DNA sequences to separate the donor and the recipient DNA sequences. Okay, then what we have is the inverted repeat elements. Okay, so TN3 has 38 base pair inverted repeat elements, as we have seen in the case of this composite transposition, the insertion sequences. Okay, same kind of insertion sequences are also found here. Okay, in the composite transposition we have both kinds of insertion sequence. Okay, so one is in the direct orientation, and the transposition 10 and transposition 5 will be having the inverted orientations. Okay, same here, transposition 3 also has a 38 base pair inverted terminal repeats. Okay, which are unrelated to the insertion sequences. So insertion sequence elements. Okay, these are not actually the insertion sequences elements. These are actually simple repeats. Okay, simple inverted repeats. Okay, and these repeats are of about 38 base pairs, and we also have a transposition gene over here and resolvase gene over here, and the beta lactamase, which is involved in the antibiotic resistance for the bacteria, which destroys the penicillin and the ampicillin. Okay, so this is the typical structure of this transposase composite transposon TN3. Okay, which also has the transposase enzyme activity, resolvase enzyme activity, and also the antibiotic resistance activity. Okay, so how does this non-composite transposon gets integrated into different locations? Okay, so first what happens is the blunt cuts are made on the donor molecule over here. Okay, like this, by the transposase enzyme. This transposase cuts the sequences of donor molecule and the recipient molecule. So both molecule will undergo the blunt end cuts. Okay, so blunt end cuts are made on the recipient as well as the donor molecule, and these are actually gets combined. Okay, so in the second stages, these single stranded cuts or the staggered, here the transposase is performing the staggered cuts. Okay, on the recipient molecules, actually the staggered cuts are made. Okay, so this both donor molecule and the recipient molecule will now get combined. Okay, so to exchange the transposable element. Okay, so when they exchange, they form an intermediate. Okay, so where the actual DNA synthesis happens on the single stranded regions. Okay, so wherever the gap is there, actually here these gaps. Okay, so these gaps are filled with the DNA bases or nucleotides. Okay, by the certain type of DNA polymerase enzymes. Okay, so this is actually called as the DNA synthesis. Okay, and during this process, an intermediate is formed, that we call that as the co-integrate. Okay, so this co-integrate is now involved in the recombination process. This is actually involved in the recombination process event. Okay, so this whatever the mechanism which is happening over here. Okay, the actual mechanism right now. Okay, so this is actually called as the recombination process, where the exchange of the transposable element is taking place between the donor and the recipient molecule. Okay, so what we call that as the recombination event. Okay, so the intermediate structure is called as the co-integrate, where the recombination events are happening. Okay, so this co-integrate will be cleaved by an enzyme called as the resolvase. Okay, okay, so normally the bacteria involves different types of recombination enzymes, like REC-A protein and REC-BCD complex, or UER-AB complex and UER-C complex, UER-C proteins. Okay, so these are the major recombination proteins which are present in the bacteria. But in the case of transposons, this enzyme is involved in the cleaving the recombination sites. Okay, on the co-integrate intermediate. Okay, so when this resolvase cleaves the recombination sites of this co-integrate, it will separate the donor molecule as well as the recipient molecule. Now the recipient has the transposable element or the composite non-composite transposon. Okay, and donor molecule also has the some part of the transposon, non-composite transposon, and again this non-composite transposon gets duplicated over here, like in these regions. Okay, like this, which indicates a copy and paste mechanisms. Okay, so recombination between duplicated transposable elements generates two DNA molecules. Okay, on the donor molecule as well as the recipient molecules. Okay, this is how the non-composite transposons undergo transposition activities. Okay, undergo transpositions. Okay, so this is what about the non-composite transposons. Okay, so remember, so these are actually staggered cuts only, not the blunt end cuts. Okay, so on both sides we will be seeing the staggered cuts only. Okay, on the donor molecule as well as the recipient molecules, staggered cuts are performed. Okay, and this conservative and the replicative transposons are quite related. Okay, so till here, whatever the cuts are made, and whatever the combining thing is there, combining thing is forming between the donor molecule and the recipient molecule. Okay, till here everything is same in the conservative process and the replicative process. Okay, but after this one, after this stage, everything gets different, or everything is different from this stage to this stage. From this stage to everything is different, in the case of this replicative and the conservative transposition activities. Because what happens is, in the replicative, what we have seen is, there is some sort of DNA synthesis happening in the replication force. Okay, and we'll be having a intermediate called as a co-integrate, where the recombination events are happening, and the resolvase enzyme will be cleaving the recombinational sites after the recombination events. Okay, which forms a donor molecule as well as a recipient molecule, which the both of them contains a transposable element. Okay, like this. Okay, but in the case of conservative transposon, which is not like this replicative mode, which does not involve any kind of DNA synthesis, DNA duplication processes. Okay, so this makes the additional cuts. Okay, additional cuts in the sense the cuts on the donor molecule over here and the cuts on the recipient molecule over here, like this. Okay, so these kind of additional cuts are made so that the donor molecule will leave from the site as it is. Okay, like this, which will not be contain any kind of transposable element. Okay, and the recipient molecule will now get ligated by the DNA ligase enzyme. Okay, and will now have the transposable element. Okay, so this is what different difference between the replicative and the conservative transposition. Okay, so the only thing is DNA synthesis or the duplication process is happening in the case of replicative transposons, that is in the case of non-composite transposon, while the the duplication process is not happening, except that additional cuts are made on the co-integrate. Okay, you cannot call that as a co-integrate, but the mixture of the donor and the recipient molecules, the additional cuts are made, and this donor molecule and the recipient molecules are now getting separated, where the donor molecule doesn't contain any kind of transposon, because the transposon is actually donated to the, or which is actually transported or inserted on the recipient molecule. Okay, so this is what the difference between the replicative and the conservative transposition mechanisms. Okay, then I've said you in the beginning of the lecture that these transposons are also involved in the mutation process. Okay, that means they can also cause insertion, deletions, and the inversions. Okay, and the major reason behind this kind of mutations is abortive transposition activities. Okay, that means the failed activities of the composite transposons. Okay, of the failed movements of these composite transposons makes the mutations or inversions or the deletions. Okay, so they result completely from the failed attempts. Okay, or the abortive transposition activities. Okay, now so consider this is a transposon, composite transposon. Okay, which has 4 sites where the cuts are made of. Okay, like either it might be the blunt ends or the staggered cuts. Okay, so these are the 4 sites where the cuts are made by the transposase enzymes. Okay, so we have here A, B, C, and D. Okay, okay, so these are the 4 different sites where the cuts are made. Okay, so A, B, C, and D. Okay, so B and C are referred to the inside ends. Okay, so these B and C are referred towards what? Inside ends. Okay, and A and D are referred to as the outside ends. Okay, okay, so whenever a transposase enzyme performs cuts at these outside ends like this. Okay, the entire transposon is now ready for the transposition from donor molecule to the recipient molecule. Okay, so that means the entire transposon gets inserted into different location. Okay, but if the cuts are made in these sequences, okay, the inside sequences, if the both cuts are made in the inside sequences, then the DNA molecule will get transposed into different regions of the chromosomes, okay, which is not actually the transposition activity, where the this element is actually not getting transported, only the DNA molecule is getting transported, transposed, okay. So this is not, this is not a transposon, and it doesn't have any kind of transposase gene, okay. So this won't be acting as a transposons, okay. So these kind of things, this kind of transpositions are also referred as abortive transposition activities, okay, or the failed attempts of this transposase enzyme, okay. Now see what I have given in these points, the two of these inside ends related to transposon, and the two are outside ends, okay. I've said you A, B, C, D, okay. Out of this A, B, C, D, A and D are what? Outside ends, and B and C are inside ends, okay. Now transposition of a single insertion element involves one inside and one outside element, okay. I've said you, and normal movement of the whole composite transposon involves two outside ends, okay. So that means these two outside ends should be cut by the transposase enzyme, so that entire transposon will get transposed into different locations, okay. And this point, so the fifth point, but suppose that two inside ends are used for the transposition, the whole of the DNA molecule outside the transposon will be moved, okay. So that means except from this transposon, okay, whatever the DNA is there inside this plasmid, it will be moved into the different locations, okay, not the actual transposon, okay. The DNA molecule will move into the different locations, okay. So this DNA molecule doesn't have any kind of transposon gene, so it won't be acting as a transposon molecule, okay. So this is what called as a abortive transposition activity, okay. So because of this, some kind of insertion mutations will be happening, okay. So this is this DNA segments, which is actually coding for a different kind of protein molecules or the enzyme molecules, is now inserted into different location, okay. So this kind of insertion into different locations on the chromosome will actually break some sort of genes on the actual chromosomes, and this will lead to lot of insertion mutations, okay. This kind of inserting a DNA molecule because of failed attempts of the transposase activity is called as the insertion mutations, which are mainly the occurred due to the abortive transposition activities, okay. Then we also have the deletion and inversion mutations, okay. So whenever the transposon get degraded because of abnormal transposase activities, when it makes the cuts over here, okay. So the transposon will leave from the plasmid side, okay. It will eject off, and whatever the DNA molecule will be there, it will be broken into two parts, okay. So if the cuts are made on the A side and on like this, okay, you will be getting two different plasmid molecules, okay. So if the two different plasmid molecule religates themselves, okay, they form the deletion mutations, okay. And if the two plasmid molecules if they religate among themselves, okay, with each other, okay, if they religate with each other, they form the inversion mutations, okay. How they form the inversion repeats, in the case of this deletions, first in the case of deletions, okay, so some sort of DNA sequences are getting deleted from the this small plasmid, and some sort of DNA sequences are getting deleted from the this large plasmid, okay. Because this both plasmids are initially they are the single plasmids, okay, single plasmid molecules, single plasmid or single DNA sequences, okay. After the abortive transposition activities, we now got the two different plasmids, okay, two different sized plasmids, okay. One is a small sized one is a big sized one, okay. So this small size doesn't contain enough genes, and this plasmid also doesn't contain enough genes, and these two will now get transported into different types of ligands, okay. And this causes the deletion mutations, okay. So that means some sort of gene sequences are deleted in this both in the both plasmids, okay. So this is called as the deletion mutations, okay, which are resulting from the abortive transpositions, okay. And if they religate with each other, okay, so this insertion sequence is getting relegated to this end, okay, and this insertion sequence or this P element, this P insertion element, okay, if it gets relegated to the this insertion sequence, okay, in opposite directions, okay, so this will cause the inversions, okay. So this is also called the inversion mutations, okay. So this kind of inversions mainly due to what? Abortive transposition processes, okay. So this is what about the mutations which are mainly happening due to this uh, abnormal transposase activities. Okay, then what we have is the mu, which is also a bacteriophage, family member of a bacteriophages, and which is also has a transposing activities. Okay, as I said you in the beginning of the class. Okay, mu replicates through so first this one. Okay, a few bacteriophage genomes reproduce by transposition and use transposition to insert themselves into a bacterial chromosome in their lysogenic cycle. Okay, so what it says is, in the initial stages, when it want to insert its genetic sequences into different parts of the chromosomes of bacteria. Okay, so it mainly perform this transposition activities in the lysogenic cycle. Okay, so whenever it is undergoing a lysogenic cycle. Okay, so it inserts its DNA sequences of the transposons into the bacterial chromosomes. Okay, and then whenever this mu is getting replicated, okay, or whenever the lytic process is happening, okay, during this lytic cycle, mu replicates by transposing randomly across the host chromosome, which can occur about 100 times, showcasing a robust transcription mechanisms. Okay, that means during the lytic cycle it will replicate itself. Okay, like by the transposing into random locations of the host on the host chromosomes. Okay, it will replicate itself. Okay, and causing lot of production of lot of bacterial viral DNA sequences inside the bacteria, which will then generate lot of mu bacteriophages. Okay, again further. Okay, and this is the typical structure of this mu bacteriophage transposon element, which has phage genes and also head and tail genes, which encode for the head and tail proteins. Okay, and we also have the direct repeats, which are oriented in one direction. Okay? So this is the typical structure of the mu bacteriophage, and the length of this is the length of this mu bacteriophage is 38,000 base pairs. Okay, and then we have the conjugative transposons, which are also referred to as the bacterial conjugating transposons. Okay, because they involve two different bacteria for their transposition activity. Okay, so majorly we have three different types of conjugated transposon, that is transposon 916 and transposon 5253 and also transposon 1549. Okay, so three of them provide antibiotic resistance for different types of antibiotics. So TN transposon 916 will provide the resistance for tetracycline, while TN or transposon 5253 has multiple antibiotic resistant genes. Okay, like ampicillin or penicillin. Okay, so these kind of different multiple antibiotic resistance will be seen on this TN 5253 transposons. Okay, and the TN1549 is majorly responsible for the vancomycin, and which is under investigation. Okay, which is now under investigation. This transposon is being studied even now. Okay, so this is responsible for the vancomycin resistance. Okay, in the bacteria. Okay, so if you look at the typical structure of this transpositive element, okay, we have the recombination specific protein genes. Okay, the excision gene and the integrase gene over here. Okay, XISINT, which is completely encoded by a single sequences. Okay, and then we have the accessory genes, which are required for the conjugation, which are required for the replication processes. Okay, and formation circularization of the bacterial genome. And then we have the genes for the conjugation processes. Okay, so this is what conjugated transposons do in the bacteria. Okay, and these are the major properties of this conjugated transposons. Okay, and majorly found in the bacterial cells, and they get transposed from one bacteria to another bacteria by cell to cell contact process. Okay, so this is what happens. Okay, so what happens is, what we have here, we have the donor DNA. Okay, and which also has the direct repeats on both sides of this antibiotic resistant genes. Okay, so here we have what antibiotic resistant genes, and on both sides of this antibiotic resistant genes, or on either sides, we have the direct repeats. Okay, and we also have a certain type of proteins inside the bacteria, that is XIS. XIS is also referred to as the excision. Okay, which perform the cuts. Okay, which cuts the DNA sequences. Okay, and INT refers to the integrase enzyme. Okay, so these are the major enzymes which are involved in the transposition activities. Okay, so what happens initially is, this transposable element is first looped into circular plasmid like molecule. Okay, and then this excision, this enzyme will be cutting this edges of this circular loops and will make the circular loops into a circular intermediate. Okay, so this is called as a small, this is referred to as a plasmid or circular intermediate, which contains an antibiotic resistant gene. Now this resistant gene will be transferred to the recipient bacteria, okay, during the conjugation process, and after getting transferred, this whole new circular intermediate will be inserting itself into the target DNA over here, okay, with the help of this integrase gene. Okay, so this integrase gene will also cuts the target DNA and integrates this entire circular intermediate into the target DNA sequences. Okay, so this is what happens in the case of this conjugated transposition mechanisms. Okay, whatever the process I have explained in the in the previous image, the same thing is given in the descriptive way in this slide, so you can just read it through. So this is about the transposons and their basic activities, majorly about the bacterial transposons. Okay, so in the next class I'll be discussing about the eukaryotic transposon, the different types of retroviral mediated transposons or the retrotransposons. Okay.